Good afternoon, dear colleagues. My name is Holubchyk D, and today I want to present to you our work titled Limitations of Classical In Vitro Models for Assessing the Hypocholesterolemic Activity of Lactic Acid Bacteria. Cholesterol is an extremely important metabolite of the human body that plays a role in regulating numerous vital functions, both at the cellular level, membrane permeability and fluidity, gene transcription, etc., and at the level of the whole organism. It is a precursor of steroid hormones, vitamin D, bile acids, etc. However, its excess leads to hypercholesterolemia, which manifests in various cardiovascular diseases that are considered the leading cause of mortality in the modern world and require comprehensive intervention, including efforts from the scientific community. Probiotic organisms, and especially lactic acid bacteria, (LAB), have considerable potential to exhibit hypercholesterolemic activity and to be used as a preventive means against the development of hypercholesterolemia. However, before the application, they must undergo multiple stages of confirmation of their activity, including in vitro, in vivo, and clinical studies. Problems mostly arise when harmonizing doses and extrapolating the in vitro data obtained to in vivo conditions. The aim of this work was to determine the main shortcomings of in vitro models used to study hypercholesterolemic activity and to identify possible reasons for discrepancies between the results of in vitro and in vivo tests. Uh, the main mechanism of cholesterol removal in vitro proposed in the literature is the ability of bacteria to assimilate or adsorb this molecule from the culture medium. Additional support for these findings is the presence of BSH activity confirmed under laboratory conditions, a mechanism of bile salt hydrolysis that is considered a major pathway of cholesterol removal in vivo. Conclusions regarding the predicted presence or absence of hypocholesterolemic activity are usually based on this information. However, lab possess additional mechanisms affecting cholesterol levels, including interactions with receptors and regulation of enzyme activity, as well as the influence of exopolysaccharides on lipid absorption in the intestine. An extremely important factor in the manifestation of hypercholesterolemic activity is the interaction of LAB with the intestinal microbiota and other LAB strains. For example, in the study by Frappier et al., Lactobacillus acidophilus CL1285 reduced cholesterol levels in vivo only when administered in combination with specific Lactobacillus plantarum strain, namely called 200, whereas administration of this strain alone led to an increase in blood lipid levels in hamsters. This phenomenon was described in greater detail by Zhao et al. In their study, they demonstrated that a strain of Lactobacillus plantarum capable of hydrolyzing only taurine-conjugated bile salts successfully reduced cholesterol levels when administered to pseudo-germ-free mice, meaning mice whose intestinal bacteria had been depleted by antibiotics. In contrast, the same genetically modified strain, capable only of hydrolyzing glycine-conjugated bile salts, showed no effect in these mice, but regained hypercholesterolemic activity when administered to animals with a normal microbiota. It should be noted that mice almost completely lack glycine-conjugated bile salts, but possess taurine-conjugated ones. Accordingly, the strain capable of hydrolyzing these compounds exhibited hypercholesterolemic activity independently of the presence of microbiota. The strain unable to hydrolyze taurine conjugates affected the surrounding bacteria, thereby promoting their cholesterol-lowering activity. The most comprehensive analysis of this mechanism to date was conducted by Liu et al. The VSH-positive strain Lactobacillus plantarum YB334 administered to mice fed a high cholesterol diet reduced serum lipid levels while simultaneously causing a significant increase in the abundance of bifidobacterium pseudolongum BPL4 cells in the intestine. This strain was more resistant to the deconjugated bile salt, cholic acid, formed after the action of the enzyme than the rest of the intestinal microbiota. The isolated strain subsequently demonstrated a strong hypercholesterolemic effect. By influencing enzymes and receptors of the digestive system, it stimulated the de novo synthesis of bile salts from cholesterol in the animals by utilizing its reserves. The newly formed bile salts were then subjected to the action of the BSH enzyme produced by Lactobacillus plantarum YB334, resulting in increased formation of cholic acid and promoting further proliferation of Bifidobacterium pseudolongum BPL4 cells. For a short period, this created a closed loop with positive feedback until equilibrium in the system was reached due to the limited number of Lactobacillus plantarum YB334 cells and consequently the limited amount of BSH enzyme produced by this bacterium. A possible solution to the described problem is the use of artificial models of the human gastrointestinal system, into which either individual representatives or a broad spectrum of members of the intestinal microbiota are introduced. Such approaches are considerably more expensive than classical ones. However, if the effectiveness is confirmed in numerous studies, they may become more widely used. At present, they are employed relatively rarely, particularly for the study of hypocholesterolemic activity, although some studies involving them do exist. For example, Ikimetol adapted the SHIME model, simulator of human intestinal microbial ecosystem, to analyze the effect of a probiotic composition on typical intestinal microorganisms, and also evaluated the influence of this composition on blood cholesterol levels in mice. The probiotics demonstrated hypercholesterolemic activity in vivo, which was consistent with clearly expressed effects in vitro. Incomplete consideration of the spectrum of bacterium effects may explain cases in which in vitro studies indicate the absence of hypercholesterolemic activity, whereas in vivo results demonstrate the opposite. However, the reverse situation is much more common. Established laboratory methodologies, which are used by most researchers with little or no modification, may play an important role here. The situation is as follows. To evaluate the ability to remove cholesterol in vitro, 100 micrograms per milliliter of cholesterol and 0.3% bile salts are added to a liquid culture medium, usually MRS. This approach first ensures the solubilization of the water-insoluble sterol in mixed bile micelles, which guarantees its effective interaction with lab, and second, simulates intestinal conditions, where bile acids also form micelles with cholesterol. However, when the studied strains exhibit sufficient resistance to bile, which they typically do, the bacteria grow well and the pH decreases below 4.0. Under highly acidic conditions, BSH becomes more active and hydrolyzes bile acids in micelles, leading to the precipitation of cholesterol in greater amounts than under in vivo conditions. This results in an overestimation of the cholesterol-lowering potential, which may partly explain the lack of positive results in studies involving living organisms. Conversely, excluding bile as a solution to the problem is less appropriate, as it would move the experiment even further away from physiological conditions. A potential compromise is the approach proposed by Bhatt and Bajaj to assess cholesterol removal both with the addition of bile salts to the medium and without them, thereby estimating the approximate contribution of cholesterol precipitation with bile salts, which will be overestimated compared with in vivo values. The accuracy of the methods themselves may also play a role. The formaldehyde method remains the most commonly used approach for evaluating cholesterol removal in vitro, although it is significantly inferior to more modern techniques, such as high-performance thin-layer chromatography, high-performance liquid chromatography, etc. Conclusions. The main reasons for the discrepancies between the results of studies on hypocholesterolemic activity in vitro and in vivo include consideration of only part of the cholesterol lowering mechanisms, the inability to adequately assess the interaction of lab with the intestinal microbiota, and the insufficient accuracy of the methods used in laboratory research. Here are references used during the writing of the work, and thank you for your attention.